Monoclonal antibodies. Monoclonal antibody is an identical immunoglobulin produced by a single B cell clone and is directed against the same epitope on a target antigen. Over 100 MABs are FDA approved and it is the fastest growing class of therapeutics. It is a Y-shaped structure. It has an AB region that contains variable domains for binding to a single highly specific target epitome, the FC, the fragment crystallizable, the effector engine that interacts with the cell surface receptors, FcRn, and complement proteins to trigger the immune response. It determines the 2 to 4 week half life. The MABs are preformed standardized immunoglobulins bypassing the need for a primary immune response. The difference between the polyclonal and the monoclonal antibodies. The polyclonal antibodies, they are heterogeneous mixture from a multiple B cell lineage. They target recognizes multiple different epitopes on the same antigen. The pros are it has high overall affinity, robust detection, tolerant of minor antigen changes. The cons are there is batch to batch variability, high background noise, and has non-specific binding. The monoclonal antibodies, they are identical immunoglobulin produced from a single B cell clone, and the target is highly specific for a single defined epitope. Pros are infinitive renewable source, zero batch variability, high reproducibility, precise therapeutic targeting. Cons are vulnerable to epitope loss via chemical treatment, expensive and complex to produce. The monoclonal antibodies are produced by the hybridoma technique. Immunize, harvest, fuse, select, clone, and produce. In immunization, the target antigen is injected into mouse and spleen enriches with antigen specific B cells. In harvest, the splenic B lymphocytes are isolated. In fuse, the B cells are fused with the immortal myeloma cells using polyethylene glycol or electrofusion. In select, they are cultured in hat medium to isolate successful fusions. The hybridomas. Cloning. Limit dilution cloning isolates a single cell to expand a monoclonal line. And in produce, it cultured in bioreactors for continuous large scale antibody harvesting. It can be remembered by mnemonic I have found some crazy protein. On the hat medium, which is used to culture the monoclonal antibodies, the unfused B cells, the H fades out, dies naturally due to short lifespan. The unfused myeloma, immortal but HEPRT negative, strikes the HAT barrier. Aminopterin blocks de novo nucleotide synthesis lacking HEPRT. They can't use the salvage pathway and the cell dies. The fused hybridoma, immortal from the myeloma plus the HEPRT positive from the B cells is combined. It successfully passes through the HAT filter, survives and proliferates indefinitely. The purification post-production bioreactor fluid is a complex soup of media components, host antibodies, and cellular debris. Extracting the pure monoclonal antibodies requires precise charge-based chromatographic separation. The first step is conditioning, in which centrifugation is done, which filters and removes lipids, clotted material, and cellular debris. In ion exchange, the leverages the specific isoelectrode point of the monoclonal antibodies. At a specific pH, monoclonal antibodies carry a positive charge, while impurities carry a negative charge for magnet-like separation. The third step is affinity purification. The monoclonal antibody selectively binds to mobilized protein A by G resins. Gentle elution buffers release a product with more than 80% purity in a single step. The The monoclonal antibodies were initially produced from mouse. Now they are even being produced from humans. The murine, denoted by O, is 100% mouse protein. High immunogenicity is present. Example is muromonab, OKT3. In the stage 2, the chimeric XI, it is 61% human. Human constant regions, mouse variable regions are present. Examples are rituximab and infliximab. Humanized, that is denoted by ZU, is 95% human. Only the complementary determining regions are murine. Example are the bevacizumab and trastuzumab. Stage 4, the fully human, 100% human protein created via transgenic mice or face display. Examples are the adalimumab, ipilimumab. The nomenclature of the monoclonal antibodies consists of a prefix, target subsystem, source subsystem, and a suffix. The prefix is unique to the specific drug. The target subsystem indicates the disease target. TU for tumor, CI for cardiovascular, LI for immunology, OS for the bone. The source system indicates the origin of the species, that is ZU for humanized, O for mouse, XI for chimeric, U for fully human. And suffix map is the classical identifier of the monoclonal antibodies. Example shown is the trastuzumab. Bevacizumab translates to a humanized antibody targeting a tumor, TU. The monoclonal antibodies, they are either the naked monoclonal antibodies, conjugated monoclonal antibodies, and bispecific monoclonal antibodies. The first one, the naked monoclonal antibodies, operate solely based on inherent properties, block signaling, flags for destruction, ADCC or the CDC. The conjugated is ADCs or the radiolabel. They serve as a target delivered vehicle carrying chemotherapy drugs or radioactive particles directly to cancer cells. The bispecific, they have a target binding site and 1 and 2. It binds to two different target proteins simultaneously. Example is blinatumomab. It binds to CD19 on leukemia cells and CD3 on T cells to force an immune attack. The 10 mechanisms of action of the monoclonal antibodies: receptor blockade, neutralization. They block ligand or receptor. Example, bevacizumab or the VEGF, vascular endothelial growth factors. Signal transduction inhibition. It triggers down regulation of receptors. Examples, trastuzumab or the HER2. The ADCC, antibody dependent cellular cytotoxicity. The FC region recruits NK cells or macrophages to kill target. Example, rituximab. CDC, the complement dependent cytotoxicity. FC activates complement cascade, forming MAC for cell lysis, opsonization or phagocytosis. MABs codes target recognized by macrophages, FC gamma receptors. The immune checkpoint blockade blocks inhibitory receptors PD1, CTLA4 to wake up T cells. Antibody drug conjugates. It delivers intracellular cytotoxic payload. Bispecific antibodies, redirected killing by tumor binding and T cell simultaneously. Complement neutralization blocks complement C5 to stop inflammation. Example, eculizumab. Cytokine neutralization, it binds pro-inflammatory cytokines. Example, infliximab and TNF alpha. Antibody drug conjugates. First step, navigation. Monoclonal antibody finds and binds to a specific tumor receptor, HER2 or TROP2. Second step is infiltration. The cell membrane engulfs the monoclonal antibody through its receptors, mediated internalization. The third step is detonation. Lysosome degrades the antibody, releasing the cytotoxic payload. Example, mptancin, MMAE. The cell undergoes apoptosis. The TDM1, Cadillac for HER2, breast cancer, and enfortumab vedotin, Nectin-4, for urothelial carcinomas. The tumor evasion, the handshake. The PD-L1 and the PD-1 handshake puts the T cell to sleep because of the exhaustion or the tolerance. Immune activation, the blockade. The monoclonal antibody, example, nivolumab or pembrolizumab, blocks the handshake. The T cell wakes up and destroys the tumor. Oncology inhibitors, anti-CTLA4. The mechanism is priming phase T cell activation. The drug is the ipilimumab, which is fully human. It is used in melanoma and often combined with PD-1 inhibitors. The anti-PD-1, the effector is phase immune restoration. The drug is the nivolumab and the pembrolizumab. The indicator is the MISH DMMR pan cancer, the non-small cell lung cancer, gastric cancer, and melanoma. The anti-PD-L1, it targets the tumor site ligand directly. Drugs are the atezolizumab, durvalumab, and is indicated in non-small cell lung cancer, bladder, TNBC, and biliary tract cancer. Durvalumab, non-small cell lung carcinoma, bladder, TNBC, and the biliary tract cancer. The clinical applications of the oncology of the monoclonal antibodies, the HER2 target, the drugs which act on it are the trastuzumab and the pertuzumab, the TDM1, and the cancers are the breast and gastric cancers. They cause the cardiotoxicity by reducing the left ventricular ejection fraction. The epidermal growth factor receptors, HER1, are acted on by the cetuximab and panitumumab, which are used in treatment of colorectal cancers of KRAS wild type. They cause an acneiform rash which indicates good response. The vascular endothelial growth factor A and the vascular endothelial growth factor receptor 2 are acted on by the bevacizumab and ramucirumab, which are used in treatment of the colorectal cancers, lung cancers, and the glioblastomas. They impair wound healing and have a high causation of hypertension risk. The CD20, rituximab, and the obinutuzumab are used in treatment of non-Hodgkin's lymphoma and chronic lymphoid leukemia. The patient should be screened for HBV activation before the use. And CD38, the daratumumab, is used for multiple myeloma, interferes with blood bank cross-match. The application of monoclonal antibodies in autoimmune and inflammatory conditions. The TNL alpha are targeted by the infliximab and adalimumab. Indications are the Crohn's disease, ulcerative colitis, and rheumatoid arthritis. It has high perioperative infection risk. Screens for latent tuberculosis are required. IL-6 receptors, tocilizumab, is indicated in rheumatoid arthritis, COVID-19, and cytokine storm. It masks infection by reducing the CRP. It rarely causes the bowel perforation. The alpha-4 beta-7 integrin, the drug is the vedolizumab and is indicated in Crohn's disease and ulcerative colitis. It is gut selective and preferred bridge to surgery in inflammatory bowel disease. The free IgE, omalizumab, is indicated in allergic asthma. It has anaphylaxis risk, requires post-injection observation. The complement C5, eculizumab, is indicated in paroxysmal nocturnal hemoglobinuria and atypical hemolytic uremic syndromes. It has a high risk of Neisseria infection. Meningococcal vaccine is mandatory for the therapy. The adverse effects of the monoclonal antibody treatments are the infusion reactions or the anaphylaxis. There is high risk with chimeric or murine. The drugs are the rituximab and the infliximab. Pre-medication with antihistamines or corticosteroids should be administered. The cardiotoxicity is noted with trastuzumab and pertuzumab. It causes reduced left ventricular ejection fraction, not cumulative dose dependent. Unlike anthracyclines, it requires baseline and third monthly echoes need to be done. In immune-related adverse effects, the checkpoint inhibitors nivolumab and ipilimumab, they cause itis, colitis, hepatitis, pneumonitis. It requires holding ICI with high dose steroids. In unique metabolic or the zinc signifiers, cetuximab causes hypomagnesemia and acneiform rash, which indicates efficacy. Denosumab causes osteonecrosis of the jaw, requires dental review, and perioperative hypocalcemia. Perioperative protocols. The drugs to preoperatively hold are the antivascular endothelial growth factor agents, bevacizumab. It inhibits angiogenesis, causing anastomotic dehiscence and fistula formation. It should be held 4 to 6 weeks preoperatively and is started after the wound is healed, that is approximately 4 weeks postoperatively. In anti-TNF agents, infliximab and adalimumab, TNF alpha is critical for granuloma formation. Severe opportunistic infection risk exists. It is held 3 to 5 weeks before the major surgery. Infliximab approximately 4 weeks. Adalimumab approximately 6 weeks prior to the surgery. Emergency IBD surgery. We assume that the patient is immunocompromised and a broad spectrum antibiotic is given to avoid primary anastomosis in contaminated fields. Vedolizumab is preferred preoperative due to its gut selectivity. Monoclonal antibodies in diagnostics and analytical utilities. Immunoassays, ELISA or the Western blot, they serve as primary detection tools due to their single epitope specificity, ensuring accurate protein quantification without cross-reactivity noise. The flow cytometry and cellular imaging. Fluorescent conjugated monoclonal antibodies tag specific CD markers CD4, CD8 counts in HIV, leukemia subtyping, allowing machines to rapidly sort and count complex cellular populations. Point of care diagnostics. The foundation of rapid testing protocols including pregnancy tests, anti-HCG, monoclonal antibodies and infectious disease screening, providing instant, highly specific biomarker detection. The advancements in monoclonal engineering, phase display and recombinant assembly, moving entirely away from animals, engineering libraries of genes to generate custom 100% human hybrid architectures in bacteria or yeast without in vivo immunization. Novel imaging and radioimmunotherapy, conjugating monoclonal antibodies with advanced imaging agents for ultra-precise disease detection, identifying micrometastasis and real-time therapeutic monitoring. Personalized biomarker therapeutics, shifting from broad receptor targeting to tailoring antibody treatments based on specific biomarker signatures of an individual patient tumor. The single B cell technology, direct amplification of the immunoglobulin genes from individual human B cells, capturing natural high complex antibody diversity that is impossible to mimic in vivo. The CRISPR or the Cas9 engineering, direct functional diverification and genomic editing of hybridomas to custom tailor physical stability, tissue specificity and toxic payload capacity. That's about monoclonal antibodies. Thank you.